Hello, and welcome to this video on recombination. At the end of this video, you will be able to explain the role of DNA recombination as a mechanism for DNA repair, explain the mechanism of DNA recombination and its role in meiosis, and explain the role of independent assortment in creating genetic variation. DNA recombination is one way that we manage double-strand breaks in DNA repair. DNA recombination is a mechanism for fixing double-strand breaks. Double-strand breaks occur when both phosphodiester bonds have been broken in a single double helix. If this occurs after DNA has been replicated, but before mitosis or meiosis, then these chromosomes, which are newly replicated, can become aligned. And in doing that, you create a template for DNA repair with the replicated chromosome. So in this mechanism, we begin with the double-strand break. The ends are trimmed back in order to create strands that can be used for complementary base pairing. There is strand invasion by the repair strand, such that a DNA polymerase could enter. In this case, the strand invasion, in this case, the orange strand is invading down here. Here's the 3' end of that strand. We can use this to extend the orange strand and use that to create a new piece of DNA. Likewise, the red strand up here could be used to extend the 3' end, creating a renewed copy of DNA that is identical to the copy that was there before the double strand break. If we look down here, at the end of that polymerase process, there's a ligation event, and the ultimate product is 2 identical DNA helices. Importantly, in this process, the orange strands that are shown here and the red strands that are shown here are identical because they were both made from the same chromosome during DNA replication. And so the product of this process is also 2 identical DNA helices. That's different than the DNA recombination that happens during meiosis, which allows new genotypes to arise. Remember that during meiosis, homologous chromosomes align. And we can compare that to mitosis. In mitosis, at the metaphase plate, each individual replicated chromosome gets its own space along the metaphase plate. In meiosis, though, instead, we pair the homologous chromosomes. M here stands for maternal and P stands for paternal. And so those chromosomes are not necessarily identical. They might have different alleles of the same genes. If these chromosomes here at the top were the maternal and paternal versions of chromosome 1, they still might have, say, dominant A on the maternal chromosome and recessive A on the paternal chromosome. They are not identical. So during metaphase 1, when those non-identical chromosomes are together on the metaphase plate, there's an opportunity for chromosomal crossing over. So in this picture, let's just assume that we have dominant A and recessive A, dominant B, and recessive B genes on these 2 bivalent homologous chromosomes. In recombination, the first thing that happens is an intentional double strand break. The cell creates a double strand break. After this, the process is largely the same. A nuclease digests the 5' ends, and there's strand exchange so that each of those pieces of DNA can be a template for the other. But remember that in this case, the templates themselves are not identical. And so in the original strand, we might have had recessive B on the red chromosome and dominant B on the blue chromosome. During recombination, though, we will be using the dominant B template to fix the damage that we created on the red chromosome that previously had the recessive B allele. Here we continue with the DNA recombination mechanism. So we had strand invasion and then DNA synthesis. This is using a polymerase. But remember that here we're using the homologous chromosomes as a template during this process. So we end up with the ligation event that happens here and creates what are called holiday junctions, named after Holiday, who discovered them. That junction obviously can't stay there forever. We've got these chromosomes kind of tied up in a knot. That Holiday junction can slide kind of across the chromosome. And so What's important here is how it resolves. In this case, you see that the holiday junction has slid all the way over here. And so when that resolves, the top chromosome will be red, and its partner will be mostly from the original red strand, but with some new blue DNA in there. And so what has happened here is that we have changed the alleles on each of these chromosomes. And the allele combinations that used to be dominant A and recessive B, and recessive A and dominant B, now also include a recessive only A and B option and a dominant A and B only option. So as a result of crossing over, we've created two new allele combinations that can go into gametes and create variation in the next generation. Genetic variation can also occur as the result of independent assortment. Independent assortment was originally written about by Gregor Mendel working in peas. But Mendel only had phenotypes to work with. If Mendel is looking in peas and we see one gene, in this case, it's a gene that codes for color. And in this case, we have two parents, one that has dominant Y genotypes and one that has the recessive Y genotype. And we mate those together and we end up with all heterozygotes. When we look at the offspring, we see a 3-to-1 ratio of yellow to green. Mendel used this to generate his principle of segregation. Now we would say that alleles of a single gene segregate into gametes. In this case, from the F1 generation, dominant Y or recessive Y could go into individual gametes. That's true from each parent, which is how we end up with recessive Y phenotypes, even when we had dominant Y phenotypic parents. Mendel's work also shows that alleles of different genes assort into gametes independently, and this is Mendel's principle of independent assortment. The principle of independent assortment can be explained by the arrangement of chromosomes in meiosis. So in this case, the parental generation has two genes. Parent 1 is dominant for both Y and R. Parent 2 is recessive for both Y and R. And this only works if Y and R are on different chromosomes. So let's assume we had a long chromosome that held dominant Y in parent 1 and a short chromosome that held dominant R in parent 1. In parent 2, the long chromosome held recessive Y, which I will draw in cursive, and the short chromosome held recessive R. If the parent with those chromosomes were to mate in their offspring, it is possible that the dominant alleles will all end up on the same side, and the recessive alleles will all end up on the same side of the metaphase plate. It is just as likely that the recessive of one will align with the dominant of the other. And there is nothing in meiosis that determines which of those outcomes is possible. For that reason, it's possible that in the offspring, we can get all dominant alleles, all recessive alleles, or all possible combinations in between. When you extend that to an organism like Homo sapiens that has 22 chromosomes, the combinations of possible alleles become enormous. In this picture, we see 3 different chromosomes with the maternal and paternal alleles shown, and all of the possible gametes that can arise. So the combinations get more extensive the more chromosomes we add. And so in the case of humans with 22 possible chromosomes, the way that alleles assort during meiosis 1 into meiosis 2 provides much of the diversity that we see in human populations. So between the recombination that can occur when homologous chromosomes are aligned in meiosis 1 and the independent assortment of chromosomes on the metaphase plate during meiosis 1, we ensure that offspring are different from their parents and that diversity exists within populations.